Good morning, everyone. My name is Matthew Cannon. I am a staff scientist at the Institute for Genomic Medicine in Nationwide Children's Hospital out of Columbus, Ohio. So first, I'd like to thank the organizers for allowing me to present this research today. This is actually my first time both at ISMB and in Montreal. And it's been honestly a really great time so far. Really enjoyed myself. So with that, today I'll be talking about some ongoing research and preliminary work we're doing into trying to improve the clinical significance of our drug-gene interaction data within DGIDB using publicly available FDA label packages. So a little bit of necessary background. So our lab, in conjunction with the Griffith Lab at Washington University in St. Louis, worked together to manage the drug-gene interaction database, or DGIDB. So this resource was first released in 2013, and since then its sole function has been to act as a data aggregator for drugs, genes, interactions, and gene category annotations, pulling data from over 40 different resources into one single computable resource and just making it publicly available for everyone to use. So I joined the project as a postdoc in 2021, but since this first iteration in 2013, its primary goal has been to make the druggable genome, that is the set of genes that are considered readily targetable by the state of modern therapeutics, make this data set accessible and provide a platform for both researchers and clinicians alike to generate therapeutic insights that they might not otherwise be able to see, or in some cases have access to. So to kind of illustrate more of the resource, this is a high-level overview from our most recent publication. We just released version 5 of the resource back in fall of 2023. Just starting at the top, again, we act as a data aggregator. We pull in drugs, genes, interactions, and category annotations from lots of different sources, some of which are shown here. This is not a comprehensive list. We pull these in as claims and subject our drug and gene records to a rigorous normalization routine. So our drug claims are normalized using the Variant Interpretation in Cancer Consortium, I think, VIC. We use their normalization resources to take those drug claims and ground them to an ontological therapeutic concept. Similarly, for genes, we take gene claims and normalize that with the gene normalization service through VIC to a established gene concept. Once we have that set of data, we make it searchable and store it in DGIDB proper. A researcher or clinician can come to the site, either our React-based client website at dgidb.org, or if you have a favorite programming language, you can send a GraphQL API query to our API endpoint. And if you're like me and had to learn what a GraphQL API query looks like, we do have documentation for that as a bit of an aside. So with that out of the way, let's talk about therapeutic context and how we can improve this in DGIDB. So in its current form, DGIDB, or rather, with the release of version 5, one of our primary goals for DGIDB has been to improve the clinical actionability of our data for use in clinical and variant interpretation workflows. So in its current form, DGIDB is able to report an interaction between a drug and a gene. And when we have that data available, we're also able to provide like an FDA approval metric, for example, some kind of notion that a regulatory body has gone through and looked at this interaction and there is some therapeutic benefit in humans. So I have an example search down here at the bottom of what this might look like. We've searched our drug encorafenib, we've hit search, and we can see a nice result set of 9 interactions across the different data sources we import. I've trimmed it down to just this top row. We have our drug encorafenib, it interacts with our gene BRAF, and this is considered an FDA approved, or rather, it's not always FDA. It's considered an approved interaction. So we've talked with a number of clinicians about this data, and while we found that it does work for some general research purposes, or in our case at Nationwide Children's, there is an exome sequencing workflow that uses this type of data. Largely, we found that it's relatively limited in its applicability to clinical workflows. And so in order to kind of improve this, we asked ourselves, what's the additional therapeutic context that we can provide this interaction that clinicians or variant interpretation scientists might find more valuable? So kind of expanding on this idea, currently, again, in DGIDB, we are able to capture notions of interaction type. So whether something is an agonist, and whether it's an antagonist, whether it's ligand binding, things of that nature. We're also able to represent its directionality, so whether something is inhibitory or activating in nature. But besides these two kind of major fields, we really do lose a lot of the physiological context of the interaction during our aggregation process. And if we're not losing it, it's often honestly buried in some of our interaction attributes fields. And if you don't know the data exists, it might as well not exist at all. So if we think about our original example search with encorafenib interacting with BRAF and kind of represent it as this kind of just toy example, right? We have drug A interacting with gene B. From our example search, we had our drug encorafenib interacting with our gene BRAF, and we saw that this was an improved interaction. So we have to ask ourselves, or rather, a lot of times as scientists, we will look at a data like this and take that approved metric right there as kind of a measure of trust that a regulatory body has come through and said, yes, the science has been done. This is a valid interaction, and there is the therapeutic relevance in humans. But from this data alone, we really don't know what that therapeutic relevance is. So what we're actually interested in, this is honestly probably a relatively naive way to show this, but we're actually interested in the stuff around the interaction, right? What was the therapeutic context what was the context that made that interaction therapeutically relevant, right? So we want to know the indications. What was the original indication for which this interaction was considered approved for? Was there a specific disease state that it's relevant for? Are there any pharmacogenomic relationships that we should be aware of, right? Does a patient have a specific expression of, say a CYP allele that may influence how this interaction would happen in the body? And then more classically, right, is this interaction only relevant when there is an activating mutation, say BRAF V600E, which is very well documented and studied. So again, from the current state of DGIB, we would run the risk of missing a lot of this context. So lucky for us, this data does exist, and there's a lot of it. So as part of the FDA approval process, we're backed in the United States, and we use kind of the FDA as our source for approval metrics. As part of the approval process for new drug applications and abbreviated new drug applications, entities that sponsor new therapeutics will have to go through the process of generating all the science, running it through the FDA, and getting everything approved. And then part of that process, they actually have to deposit all the label data in this publicly accessible website, or database, excuse me. So I have here an example of what one of these labels looks like. I am not endorsing Braftovi. This just happened to be a very convenient example to show. So we can look at our label, right, starting at the top, and we see our drug of interest from our previous search, encorafenib. We see that it's a capsule for oral use. We see it was initially approved in 2018. And if we scroll just a little bit down, we get to the indications and usage section where we can see pretty clearly some notions of context, right? I've highlighted here, right, starting at the first bullet point, we see that this therapeutic interaction was approved for use in the specific situation where there's also a combination therapy present, right? And that was definitely not something you could guess from just our original data set. Similarly, as I kind of alluded to, right, it's only therapeutically relevant when there is that BRAF V600E activating mutation. Again, another data point that you might have missed just from the way we currently have it represented. Interestingly also, right, our interactions with just a single drug A interacts with drug B, but we have 2 bullet points here, and there's also another indication in which it was approved for, in which there's another combination therapy it's relevant for, also with that same activating mutation. So what we might have just taken at face value as a single approved interaction could actually have 2 completely different therapeutic contexts that may be relevant for whatever we're studying or whatever our clinical workflow is. So we would like to bring this data into DGIDB and find a way to represent this for those clinical and variant interpretation pipelines. So one approach that we're exploring is the use of natural language processing. So I've been sitting through a lot of talks this weekend, and I probably don't need to talk much to this audience about what NLP is, but my graduate professional training was kind of like wet lab, preclinical development based. So I've actually been doing a lot of learning about natural language processing this past year. But briefly, NLP is a field of computer science and linguistics that focuses on manipulating human language from natural language data sets. And there's a lot of key tasks that you can perform with NLP. So I have a nice kind of cloud here that I found from a public article that kind of illustrates all the number of tasks that are out there, right? There's feature extraction class, there's classification tasks. The hot button topic these days, right, is everything generative. There's a lot of generative tasks, and there's a lot of apps out there trying to put generative into everything, even if it doesn't necessarily need it. For our case, we're more concerned with kind of this bottom right quadrant. We're looking at knowledge extraction, or rather entity recognition and relationship extraction, right? So we want to be able to perform name entity recognition, look through a label, identify, relevant chemical or therapeutics, identify genes, and identify disease or indications, or rather disease states or phenotypes that may be relevant. Once we can tag those, perform relation extraction and kind of qualify what that relation actually is and how it relates to the parent therapeutic. So I have, again, the label for Braftovi, not an endorsement, just a very convenient example, but I've annotated it a little differently. I have colored, I've represented different entities that we might be interested in with different colors. So starting with blue, right, we have all mentions of therapeutics or drug terms. So we have encorafenib, we have the brand name, we have the other combination therapies, binimetinib and cetuximab, all highlighted in blue. We have notions of phenotypes or disease states, metastatic melanoma, metastatic colorectal cancer, again, all data points that may be relevant for a clinical interpretation workflow. And then similarly, right, we have our gene BRAF, and we have that activating mutation, V600E or V600K, shown in green and yellow together. So, right, we want to be able, or rather, I've done this manually to show you this, but we want to be able to use NLP to pull this in and represent it in DGI. So I have today a preliminary workflow that we've kind of implemented to look at this to see if it's possible. Starting at the top left, right, we start at our drugs at FDA database that holds all this information. There's a set of over 200,000 labels. You can access this as PDF or free text if you want to, but I do not recommend it. They do also have a nice JSON format that you can kind of just section out the different label headers as key value pairs. Granted, there's no guarantee that once you get your relevant section, it will be human readable because they kind of vary in quality greatly, but it is there and it's very easy to access. So for this first run through, right, we took out 3 sections in particular. We took out our brand name and our application number. To be able to link these labels back to ontologically grounded therapeutics in DGIDB. And then we took the clinical pharmacology section as kind of our first kind of test look to see if this is something feasible that we could do. The reason we chose this section is because, right, if you find a genetic entity present in clinical pharmacology, let's say you say a CYP enzyme there, it's not too far a leap to infer that there might be some kind of pharmacological relationship between the use of the therapeutic and the presence of a CYP enzyme. Granted, when we would do this formally and present this data in DGIDB, we would have to nail down that relation extraction task to validate and qualify what that relationship is. So we took those clinical pharmacology sections and we found we were able to identify a BioBERT model that had been fine-tuned for almost exactly this task. It's well described. I have the link here. We were able to perform name entity recognition using this and kind of extract those entities from the text. Once we had that set of entities, we again used our Vic normalizers to go through each entity and try to normalize it to an established concept, right? We don't want to display anything unless we can point to something that's an established concept. Once we have that set, again, I kind of already alluded to this, we take our brand name and our application numbers and use that to link the parent label back to the therapeutic. So at this point, right, we have our set of entities that are hopefully ontologically grounded, and we have our set of labels that are linked to a therapeutic in DGIDB. And the idea would be to basically kind of combine these two together and have some kind of annotation of the clinical context. And so today I would love to be able to show you data on relation extraction and be like, hey, look, we did it, but we are still working on that part. What I can show you today is data on our ability to normalize these entities once they are extracted from the text. So on the left, I have the quantification of the entities we were able to extract from this set of pharmacogenomics sections. Starting with chemical on the left, we were able to extract, sorry, I'm looking for my notes, 1,327 chemical entities, 257 disease entities, and 2,515 genetic entities. When we ran these through our normalization suite, we were able to normalize between 51 to 86% of these, varying or depending on the type of entity, with therapeutics having the highest rate of 86%. For this first test run, again, we're looking at the pharmacogenomics section. We were most interested in what those genes we're actually pulling out are. might have the pharmacogenomic relationship there. Nothing too surprising here. We were able to see lots of mentions, as expected, of CYP enzymes that, again, may have some kind of pharmacogenomic relevance for the patient. And these had a high rate of normalization. It's worth mentioning, you'll see in the middle of this data set on the right, we have notions of star alleles, and these do have ramifications for use in patients depending on their expression and depending on the particular therapeutic. We did have trouble normalizing these, and there were quite a lot of these present in the set. You'll see lots of different representations for these, right? We have CYP2C19, but we also have notation for star 1, we have star 2 and star 3, and then sometimes we see things like star 2 and star 3 all. So I think if we were to kind of revisit this, right, we would be able to improve our normalization rates for these genetic terms by kind of accounting for these kinds of terminologies and different representations. So we were able to normalize our entities. We next looked at our ability to link the parent therapeutic to something in DGIDB. So using, again, those application number and brand name sections, we were able to link approximately, or rather, of the 4,099 extracted entities, we were able to link back 1,793 of these across 24 unique therapeutic concepts in DGIDB. And I have these concepts, or these therapeutics rather, shown on the right. So, for example, drugs like rosuvastatin, metoprolol, celecoxib, and there's another one I highlighted, but warfarin, right? These are all drugs that have well-described pharmacogenomic effects in humans. And as you might expect, we're able to pull out a large number of entities and attach them to the labels. So my conclusions for today, right? I would love to be able to say that we've figured out the relation extraction task, but that is something that I'm still very much learning and figuring out how to best do. I think it's likely that we'll have to fine tune something on our own to figure this out. But at least for today, we've been able to show that we can perform this name entity recognition task using what's currently out there, and then also normalize it using our normalizers to ontologically grounded concepts. And again, that rate was anywhere between 51 to 86%, depending on the entity type. And then we also showed that we were able to link these entities, we were able to link their parent therapeutic back to records in DGIDB. And so future directions, of course, as I've kind of already said, figure out that relations extraction task so that we'll be able to formally define what the relation between the section and the entity is, and then make it presentable for the researcher. Improve our normalization rates for things like CYP enzymes that have different representations of different star alleles. And then once we figure out this process, see if we can kind of explore other labels or data sets to be able to pull in that clinical context and make it accessible for clinicians and variant scientists. And so with that, I think I may have went a bit over. I'm sorry if I did. I'd like to acknowledge the help and support of everybody at Nationwide Children's Hospital and the McDonald Genome Institute for their continued support on this project.
This is super cool. I'm really excited about exploring the data set that you have. I'm wondering if you looked into using the Eliminating the Druggable Genome Project and also the FDA's adverse events.
I'm sorry, could you say the first one again?
The IDG project from NIH from a couple of years ago now. And I'm wondering how the two data sets might play together. Yeah. And then the other one is FDA's adverse events data set.
Right. So we have not looked at those sets yet, to answer your question. That is not to say that we definitely would not be interested in those data sets. This is kind of our first work through with what we saw as kind of, the first place to go for this type of data. I will say from experience in working with this, if there is a better data set to use, we would love it. The FDA labels span quite a length of time, and the quality varies wildly. And it would probably save us a lot of time and headache if there were an easier way to grab this data.
Very nice talk. A quick question. So I'm not very too familiar with the FDA labeling on describing the drug-gene interaction. Can you give a quick overview on actually mainly what's the action type? It's kind of more PK or PD or just a more safety concern. And for the database, it's more focused on oncology or this across all therapeutic area?
So I'm sorry, is your question about the drugs at FDA database and what they have, or what DGIDB has?
Your database have, it's more like, associated the drug gene interaction derived by pharmacokinetics, drug exposure relationship, or more like drug dosing, like, pharmacodynamics effect.
So DGIDB has just the notion of interactions between drug and gene. The drug at FDA database, right. So again, as part of the approval process in the United States for that, the assumption is that the entity or corporation that's sponsoring the new therapeutic will provide the research that includes things like pharmacodynamics, pharmacogenomics. Really, again, you got to think this from their perspective. They're trying to get their drug approved for use on market. So it's in their best interest to provide as much data describing the efficacy per some indication. And so, hopefully that's all available through that website.
Thank you very much.
Yeah.
Any other questions? Please.
Thank you for this great talk. Are you also extracting information from the metabolites of drugs if they interact with certain variants as well, or
So
maybe this information is not in the FDA labels? I don't know.
So I don't think I know for sure enough to answer that question. If the question is with regards to if there's metabolite data present in the labels, I think that might depend on if they provided that information, and I don't know offhand if that exists. But it might be something interesting to also pull in as a separate thing. Maybe we have researchers who use DGIB who are interested in that type of data, and maybe we could find a way to aggregate it.
Okay, thank you.
Yeah.